being here also. This presentation is going to be really a summary of my PhD, of what I've been doing, what project I've been working on. It's been going a little bit off track sometimes, but I hope I managed to pull everything together so that you can understand what I've been doing the past 3 years, essentially. This slide is going to take a little bit to load, I think. This is a bit heavy. Maybe. Okay, nice. I would like to start with this tree. This tree is Pinus sembra, also known in German as Zilber. And as you probably know, it's a kind of like endemic tree of the Alpine range. And it has been very important for several reasons throughout the history. Some key characteristics of the tree are the fact that it can resist to very low temperatures, up to -43 degrees. And so it can also grow to high altitudes, so high to almost 3,000 meters in some parts of the Alps. It grows on a very poor substrate, very poor soil, so it can grow especially in between the cracks of rocks and very close to glacier for fields in environments where no other tree could otherwise survive. It can live up to 1,000 years. And especially here in Innsbruck, we have the Zilbenbeck on Pacekofer, and there you can find a tree that is 700 years old. Of course, the location is not known for obvious reasons, but just to make you understand that this tree is indeed part of our environment, part of our everyday life. In fact, it is well known for his very social properties they can give to a party, like the Zilber schnapps, but also has been used throughout the history for other reasons, such as construction, furniture making, because it's wood, it's very dense, it's very resistant to the outdoor, and it's been used mainly in the past, especially around Tyrol, and Switzerland, for the construction of inside of arms, of huts. There was some stories saying, oh, but all these huts are very quiet, very nice, they're very warming. Indeed, only recently researchers found out that this tree, the wood of this tree emits some volatiles that relax the people that breathe it and animals around. So it has some truth. Of course, as everything that we touch, eventually we touch it too much. And so by the beginning of the last century, Zilber forests have been used for carpentry and essentially they've been deforestation all over the Alps, to the point that there were so few stands, natural stands left across the Alps, that it became a protected species. No one could touch it anymore because there was a fundamental problem. Once you cut the tree, they will not be able to grow it again. They will plant some trees, and then after 5 years, the tree will die off. They didn't understand why. And out of a huge Swiss effort to replant and reconstitute these trees, this forest, out of 5 million individuals, only 20,000 survived after 10 years. The tree was going to be extinct. So it was protected until the middle-50s. Naturally, this has repercussions, not just because it makes locals around the Alps very sad or skiers very happy because you can ski on these slopes, but it also has some huge implications for land erosions, for avalanche protection, and naturally for the sustainment, for the conservation of a very endemic tree. So in the mountain Alps, I will set aside this story, how this is going to end. I want to jump on the Alpine ecosystem because this is where not only we live, but this is the environment that I've been focused on mainly. And there are a few characteristics, essential characteristics to this environment. First of all, it's heterogeneity. There are lots of features and there are lots of exposures, and so the climate is very different in many locations, in many different parts, which makes it extremely heterogeneous and variegate. And so it becomes a hotspot of biodiversity with all these characteristics. Not just above, but also below, we see that the soil that we're standing on is not just soil. Soil is can become a very philosophical concept to discuss and to define, but essentially it is the mixture between solid components and the architectural space that they form. And this they can become very, very heterogeneous because you don't have just homogeneous material, but you have a solid phase, a solution phase with water and solutes inside, but also a gaseous phase where air is present. And of course, from a microbiological point of view, this is very important because all in a sudden, all these, you're not just dealing with a very complex ecosystem, you're also dealing with a very complex environment. And so the microbes that are microbes per se because they are microscopic, they really live in this heterogeneity of space. And this heterogeneity of space is a concept that is very important because it's like if someone had to sample a part of the United States and then they say, the population is spread, there are 10 million people per I don't know, 10,000 kilometers squared. And then you say, okay, but humans are not spread equally across the United States. There are cities, there are more densely populated countrysides. And so often in microbiology, fortunately or unfortunately, we can only predict so far. We are limited by the technologies nowadays on how precise we can actually say the reality that we are living in. Where do I step in? In my PhD, I essentially wanted to focus on the microorganisms, and you can do this in many different settings. And essentially, these settings were, among others, the majority of settings looks like this. Like you have a forest at the bottom at lower altitudes, and then you have a grasslands at middle high altitudes, and then at the top you have a very rocky surface. Hopefully, well, not so long ago, not so long before, but with glaciers on it. And of course, this setting it wasn't a key point of my PhD, but naturally, climate change has an effect on everything. So, although not directly studied in my PhD, it's something that I have to take into consideration when putting together all these variables. And so now I constructed a PhD concept where I try to study the heterogeneity of this environment and its components, its microbiological components, in as holistic vision as possible. And so essentially, there are many other microorganisms in the soil, such as viruses, archaea, protists, and many invertebrates that are very important for the ecosystem. But somehow we focused on fungi and bacteria because they're the most well-studied, the most well-known, and plants because they, of course, influence the environment so drastically. And so I put these environments that I focused on, somehow they are in between this concept. And I will start again with I will continue again with this, with the forest, with the Pinus sembra forest and Larix forest, for example. But to end this discussion where all this went, or where all this started, it started from my grand supervisor, essentially, which was Professor Moser, was the supervisor of my supervisor. And in the early 50s, he was asked by the University of Innsbruck, by other forestry department, to solve the Pinus sembra problem, why we couldn't grow Pinus sembra anymore. And essentially, what everyone could see is this. You go walk on a wood and you see trees. If you're particularly careful, you see different types of trees. You have a bush at the bottom. And then if you are even more detailed on looking, then you see mushrooms. And you say, oh, okay, there are different types of mushrooms. Sometimes these correspond to that specific type of tree. And okay, very interesting, but what can I do with it besides a good pasta or whatever if you want to make with it? If you go even deeper under the ground, how these mushrooms and these trees are connected. Essentially, these mushrooms, what we call mushrooms, what we see above ground, they're coming from somewhere. This coming from somewhere comes from the roots of the tree where they are usually around. And so how do they look? They look like this. Essentially, they are filamentous fungi, and so, which is called hypha. And this hypha, once in contact with the root tree, they form these specific structures, which are called ectomycorrhiza. Ecto from outside, myco fungi, rhiza from roots. And these structures are the fundamental answer to the problem. These structures, together with the net that the fungi forms, not just between the tree and the soil, but also among trees and among different trees, among different plants, this allows the plant to form these structures and also to grow better, because now, in a sudden, the tree is better equipped to absorb nutrients from the soil, a poor soil like in the alpine environment, better than without. And so he was the developer of what is called inoculation. So if you take soil from a forest that has trees, adult trees, and you use this soil to grow seedlings, the tree grows better. And then you put this in a new environment with these inoculants, and the tree grows. And then the Pinus Selmer forests weren't endangered anymore from the 60s on. This also raises a very interesting point, which is another interesting point, and that is the interconnectivity of woods. What is being developed, what is the theory behind, is that the proposal was that there is a wood wide web, that all these trees, all the plants even, are interconnected with each other. And so you see another tree that transfers the nutrients to a tree growing on a rock or in a different, harsher environment, and so that the resources are now shared among the whole community. So can we now talk about individuals or a whole ecosystem, a whole forest? And this is a very interesting question. Naturally, this was the 50s, and since then, essentially nothing has been done on this. There's been a lot of research based for reforestation. There are some terminologies here. Afforestation is when a forest has been cut. When there is no forest, a forest never existed on a land, on a valley, and then you want to add a forest. Reforestation, when you cut down a forest and you want to restart growing a forest there. So for these reasons, of course, there's been a lot of research, but what was the standard diversity of these fungi in a natural forest has always been off the topic. And so this is where I come in with a combination of molecular, modern, and classical analysis, morphological, where you literally take the root and you see and you describe it. Of course, there is climate. So in this sense, you can think of natural stands as one valley being warmer than another valley. For example, Finchgau, where the picture was taken at the beginning, that's a very warm valley compared to another valley in South Tyrol that is a north exposed, and so it's colder. And now you have a natural environment that simulates the climate change, if you wish, to some extent, of course. And then, of course, there is a discussion about season and age, because it's not always the same. As degrees grows, it's going to have different community partners. And so these are essentially the questions that I try to answer to Pinus Sembra forest and Larix deciduous forest. But for the sake of time, I will only explain briefly this one. So do we see between a warmer and a fresher environments, do we see the same mushrooms, the same fungi, or it's going to be different? And it's almost self-explanatory. What is this? This is a graph that essentially takes into consideration abundances and diversity. So what type of mushrooms you have on the root of a tree. And then it tells you, okay, in this location, you have this certain amount of certain types of mushrooms. In this other location, you have different things. And then how comparable these two are. The more comparable they are, the closer the dots are. The less comparable they are, the more distant they are. In fact, it's a distance matrix-based approach. And you can see, it's very clear that in the Pinot-Sembra forest, the community is very location-specific. And whereas in the Larix decidua, there is essentially no pattern. So everything is everywhere and is much more dynamic, the diversity. This has implications because when you want to plant a tree, then you'll know, okay, I really need the inoculation or the mushroom from that location to be successful, whereas not is the same for another tree. So not all the trees are the same. And this was kind of like an interesting point. From these results and the research question, 2 papers came out. One is already published in Pinot-Sembra, and another one is just about to be submitted. Now, we talked about fungi and plants, but what if we add an extra layer of complexity, bacteria in the soil? How this interaction is gonna be affected? And so to do this, it's already complex enough that if you go on the ground, in the field, it can be hard to manipulate and to control for factors. And so we made an experiment. But before going to the experiment, there are some extra complexity that I need to explain you. Besides ectomycorrhiza, we also have another type of fungi that is called arbuscular mycorrhiza, which in this case, they do essentially more or less the same things, except that their life cycle is a bit different. They are very important for the adaptation of the plant to specific environments. They do colonize the soil, and they strongly have interaction with the microorganisms around the roots, which are called rhizobiome, the biome around their roots. And so they essentially have 2 life cycles. One is when originally they are like this in the soil, they are spores. They don't make mushrooms, they only are spores. And these spores at the beginning of the season, spring usually, they get chemically activated, and they can in fact colonize the plant and improve their nutrition, especially for water and phosphorus, but also for defenses against pathogens and stuff. Another important member of some plants are rhizobia. Probably this is a bit well known because they form nodules, and these nodules are essential for nitrogen fixation. So the air is filled with nitrogen. These guys can fix this into soluble forms, and so enrich not just the ground, but also the plant, because they provide direct nutrients. Of course, these 2 types of symbiosis, what is called, these collaborations, are costly. They cost energy in forms of carbon, fixed carbon, sugars, essentially. So now, what is the experiment? We took a plant, a representative plant of the grasslands, which is Trifolium protense, or red clover. As you probably were walking in the summer on a ski piste, on a ski slope, whatever you are stepping on is 80% this guy, Trifolium protense. They are loved by cows and also from some kids. I don't know, probably there are some quadri if you're lucky. I haven't seen any in this. So anyway, we put them in pots. We controlled 2 variables. We wanted to see what's the effect of reduced numbers of microorganisms in the soil, and then we applied drought, simulated climate change, extreme. So you can see that the water content was nearly 0, 2 parts in some of the plots. And this was a very nice collaboration, because I'm definitely not an expert in plant physiology, but my colleague from the ecology department studied the above ground and the below ground plant traits, so whatever components that the plant is done, you have to deal with, because remember, this is we wanted to see the whole picture, really, what is happening to everything in this system. So she dealt with this, and I dealt with the microbes. And so, from the collaborations of our two efforts, well, I already explained the novelties. What was the results? Essentially, I will focus on the microbial side, but of course, this is a complex picture, so a little bit I will have to tell you about the whole story. Essentially, in native conditions, drought or no drought, there was no effect on the plant physiology. So, as expected, some types of microorganisms reacted well, because under drought, the plant needs more help to get water. More water, the need of more water means more symbiosis, more collaboration with fungi and bacteria, and so they go up, especially in the drought. These are the arbuscular mycorrhiza fungi plot, and so they are higher, and also here, they're higher than in the moist. But when we reduce the microbial load, when we try to remove as much microorganisms as possible, then the story changed, because now on your side that you have a lot of productivity, but the plant acclimatized in a different way. So the plant traits, leaf number, leaf surface, how long the roots are, stomata on the roof, respiration, all these things changed dramatically. And although the microorganisms were still very present, we see a response in rhizobia, where the plant has to diminish the number of nodules in the roots so that it can copy with both the stress and the amount of resources that it's giving away to collaborators. What this means overall, it means that the system is balanced when it's whole. Take a section part away, even if you reduce the pathogens, pressures, or the parasitic in the roots, the symbiosis, the good guys, are not enough to equally sustain the system. The plant adapts, yes, but together with these adaptations comes with costs and also fitness. So how likely you are to then sustain another season. This was the result of two papers, again, one from their side, which is more physiology, plant physiology, and another one that I just submitted, which is about not just the abundance is important of the microbes in the soil, but also the diversity. So this all really, you can never take off a piece of the system and be satisfied with your answer. You really have to see this complexity as a whole. Otherwise, you're missing something. Lastly, what happens if we take off a complexity, a layer of complexity? What if we take off the plant? Now we talked about fungi and plants, fungi, bacteria, and plants, and now I want to talk about fungi and bacteria. And to do this, I will have to go in an environment where is, of course, without plants. And if you can think of any, that's a glacier force field. Now, I would like to ask how many of you have ever been in a glacier force field? In any time. Okay, few subjects. Okay. For the other subjects, I will show you this. And then you will be able to redefine your answer. So this is a map. This is a model developed by some guys in the glaciology department, where they model the dynamics of glacier covers of the Alps. And this can see we are here, and 40,000 years ago, we were under ice. And then again, it diminished. And this goes very well along with the temperature, the black one, and the amount of ice volume. So the lower the temperatures, the higher the volume of ice. And this you can see that reaches almost to Munich. And this is so of course it's a model, but I think it proves the point that everything at some point in time in the Alps was a glacial field. Nowadays, almost nowadays, 200 years ago, this is a picture from a glacier in Hallstatt, in Dachstein complex, between Styria, Tyrol, and Salzburg. And this was taken from the first alpinist who ever got there. And he walked on the glacier, got there, Simone Hutte was born, the rest is it. Today, we had to walk 3 hours to get to the base of the glacier, and then another 5 hours to go up the glacier, and up the Hutte, and to ski down to our sampling site, which was around here. It took great effort and courage from my side, but my supervisors were just fine. And so once we got there, we essentially took some soil samples. I already said why glaciers for fields. Naturally, they're well documented in the Alps. So there's a social interest, cultural social interest. I already said they're extremely simplified environments. But one extra point that I would like to make is that they provide defined age classes. It means that 20 years ago, the ice was here, 40 years ago was here, and so on. And this tells you how old the soil there is. Okay? So for our scope, we wanted to focus on the very, very early developed soils. I don't remember the next slide. Yes. So maximum of 25 years of ice-free. It means that if you go there 35 years ago, it was ice. It was nothing. It was not exposed to the atmosphere. And so now we wanted to sample these environments, looking for bacterial and fungal interactions. And a particularity, not so important now, is that these 4 glaciers that we sampled have calcareous bedrock, which means it's different from the majority of studied glaciers that have a silicous bedrock, and there is differences in soil conditions and other complex mineral stuff. So we sampled 4 glaciers because it's important to know that these environments, although similar, they're extremely complex because they are covered by snow 9 months of the year, and then the other 3, they have they're extremely dry, a lot of UV, but then it can precipitate a lot, can rain a lot all in a sudden, and there is a lot of mineral weathering. And so in the winter, it's covered by snow, so the temperature doesn't really go below 0 under these 8 meters of snow. So they are very complex, and there are mudslides, avalanches, lots of things going on. So we needed 4 sites to replicate our results. It comes naturally to think, okay, you have 4 sites. Can we find, with these very high turnover rates, which is essentially this complex dynamics, can we find a core microbial community? Which are the bacterial fungal interactions at this site? And what drives the microbial community assembly? How do these populations come to be? What are the drivers? I will focus mainly on this second point again. This is a little bit an overview of what happens. 10 plots, each glacier. This is the ice, equally spread across this area. General geochemical parameters measured. And now for the sake of understanding a little bit better the methodologies that brought me here at the DiSC, I will try to explain you how microbial ecology is working with soil samples or samples in general.
So we said we have two populations that we want to communities that we want to target, and essentially these two can be boiled down to containers, cells. Okay. Inside a cell there is DNA. Yeah. Okay. DNA is essentially a combination of 4 letters, nucleotides. Okay. And depending on the combinations, it has different repercussions on the cell, right? But also you can use it as a number plate. Okay. Inside the DNA of every single container, there is a number plate, and this number plate is named a specific way. For bacteria, it's 16S, V416S. For fungi, is ITS2. Whatever. These are practically identifiers of each cell. Okay. So then you go there, you extract these cells from the whatever matrix you have. You break these cells apart, you extract the DNA, you sequence this number plate, and what you eventually end up with is a big matrix where on the rows there are wherever the number plate come from, so the soil samples, so the locations, location 1, 2, 3, whatever. G means Grissenfirn, one of the glacier, M another glacier, Marmolada. Each number plate is somehow similar to each other. It's just like saying, okay, now cards here starts all with I because it's Innsbruck, and cards in Salzburg, they start with S. And so a way to combine and reduce somehow the diversity and the complexity of this environment is like, okay, bunch together everything that starts with E and everything that starts with S. And so by a parameter that we define, there is a 97% similarity of these number plates, and then you end up with cards. Okay, with each one of these is a microorganism. Each of these microorganisms has been found 0 times in these samples, while this one, for example, was pseudo-absent in G, but more present in M. And so you have each one of these, you can reduce every single number plate to a count, so you have 30 individuals, or 54. And this matrix gives you an overview of the diversity of what you have, the diversity, abundances, numbers, and also the occurrences, where each of these microorganisms appear where. Nice. Then you run many lines to assure that this is sound and it's clean and doesn't have too many biases. And then you can start playing a little bit. Each question that I showed you before had had many different approaches, of course, 3 different methodologies. I will not go into detail of any of them really, just the network analysis, which is essentially what it is. It's you try to predict based on abundances and occurrences who is with who. So we are now here in the same room, so we will speak all together. Essentially, we will create a nice network. But someone that is in another office will not appear here because he's not interacting with us at this specific moment in time. And so you have bacterial and fungal interactions, where each of these symbol, it's a microorganism, one of those OTUs, and these lines connects them. So it means that there is one, for example, this one I know, that is connected 4 or 5 times with each other organism. And then we can see who is interacting with who, essentially. Other important things to know is that we wanted to predict which are the bacteria-fungal interactions, how do these networks look in each glacier, but also we wanted to see if there are common associations. Can we find 2 microbes that interact in these 4 glaciers? The premises is that we did find a core of microbiome, so individual number plates present in different glaciers. We found that. And now, to obtain to see if we have common associations, we essentially overlapped them, and we found none. But is this number expectable or not? And this is also where it comes a little bit of computational power, because essentially what we do in microbiology is we see a reality, and then we try to see, okay, this reality is very nice, but is it different from a random distribution of the data? And so random distribution of the data means that you try to do this to predict this association as many times as you can. And so we did it. So we first had the parameters of each of these networks. We knew how many associations there are. And so we calculated 900 times by shifting their abundances or occurrences to reobtain the same number of associations. And then for each of this time, we saw how many there were in common across networks. Okay? No, we should have not expected any associations, which it sounds easy or simple, but if you think about a purpose of a scientist or a researcher is to try to find meaning, and somehow this meaningful view of the world has the upper leverage on any publication. When we find something that is not expected, it has philosophical implications, and also it means that the environment does not select for specific associations. And so whatever is there is gonna interact with different members for survival, but these interactions are not predictable. And so there is a lot of contingency, so stochasticity, randomness in the system. And this is then when we evolved to the third hypothesis, third question, that we wanted to see what is driving this stochasticity, right? I will not talk about it, but this is more or less what it means. Collaboration, this is also thanks to Miguel, and we just submitted this paper. Yeah, so waiting for their response. Yes. Now, the way forward. All these that I've been talking about is essentially based on a section of possible methods in microbial ecology, but there is also another part, which is laboratory work. And essentially, I did a review on how to eventually reconstruct the complexity of the soil in the lab, and then to different methods where we can see and we can measure the actual interaction among them. And it's not predicted, but some experimental. And there are two ways. There is a physical interaction, of course, and there is a molecular interaction. And this was also the result of another publication that is already outside. In conclusion, I will just a little bit draw back to the human side of the story, which is what I learned out of all these projects. And essentially, I could boil down to 2 or 3 points. One is that the project design is very important for obvious reasons, but coming from not having background on this, I found it particularly important, and I struggled particularly with some moments where our old work was put in risk because of a fundamental bias in the design of the idea. So this is essentially where I think most of the effort should go at the beginning. Another important point that I found out is that there is a lot of data out there, and there's a lot of data you can get out of it, and then you have to go through it. And this is really complex. I found it particularly complex because I don't have this bioinformatical approach to these big data sets. And so for me, it was very important to do one step after the other one, and having in mind really, okay, what do I want out of this piece of analysis? And then, as a final, as a lastly interactive result that I learned is that it is very nice to ask for help and for come around to the scientific community and share your ideas, share hypotheses, share analysis to then eventually come up with a more solid and easier way. Okay, with this I finished. I thank you for the attention, and I would like to thank particularly my supervisors, my group, Miguel, and all a bunch of other people that supported me and helped me with analysis. Thank you.